Today we're going to be learning about the chemistry of bioluminescence. Let's start by defining bioluminescence. Bioluminescence is the biochemical emission of light from a living organism. Bioluminescence can be found in a variety of living organisms, from bacteria, such as the Vibrio fischeri species, to mushrooms, such as the Pinellus stypticus species. Bioluminescence is most commonly found in marine creatures like the deadly blue-ringed octopus or the deep-sea anglerfish. Now let's move on to the purpose of bioluminescence in living organisms. What could be the advantage of an appetizingly glimmering mushroom or glowing fish? Wouldn't bioluminescence make the organisms stand out, making them more susceptible to predators? So far, scientists have confirmed three main evolutionary purposes for the existence of bioluminescence. The first evolutionary purpose is for predatory offensive functions. For example, predators like the anglerfish can use the glowing bulb that protrudes from their back and dangles in front of their mouth to illuminate their prey. Anglerfish live in the depths of the Atlantic Ocean, where there is no light from the surface of the water. This leaves the marine organisms in pitch-black darkness, so anglerfish have a major advantage by being able to produce their own light. Predators can also use flashes of light to stun their prey. The second evolutionary purpose is for defensive functions. Similar to how predators can use flashes of light to stun their prey, some prey can use flashes of light to confuse their predators, allowing them time to escape. Bright lights can also signal an animal's dangerous nature, a warning to predators to back off. The previously mentioned blue-ringed octopus lives in shallow tide pools and coral reefs in the Pacific and Indian Oceans. The octopus will put on a threatening display of its bright blue rings when it feels threatened. The octopus is one of the most venomous creatures in the world, and one painless bite is fatal. The third evolutionary purpose is communication. Fireflies use their bright lights to communicate with each other and to find mates. Bioluminescence occurs as a result of a chemical process that takes place within the bodies of the living organisms. This chemical process is an oxygenation reaction between a substance called luciferin and oxygen. Luciferin is an organic substance that is transformed into an oxyluciferin through a series of oxidation-reduction reactions. Luciferins are unique to different organisms. For example, dinoflagellates, which are single-celled marine plankton, have a luciferin with a chemical composition of C33, H38, O6, N4, and A2. Well, the luciferin that is responsible for a firefly's glow has a chemical composition of C11, H8, N2, O3, S2. This oxygenation reaction is catalyzed by enzymes called luciferases. The diagram towards the top of the screen demonstrates the oxygenation reaction by which luciferins produce light. The diagram towards the bottom of the screen demonstrates the lux operon, which regulates the production of the luciferases. Quorum sensing is the process by which gene expression varies based on cell population density. A common example of quorum sensing is the bobtail squid and the Vibrio fischeri bacteria. The bioluminescence of a single Vibrio fischeri bacterium would be a waste of energy. So, the Vibrio fischeri bacteria wait until the cell population density is great enough to bioluminesce. The bacteria can sense the population density of the cells through autoinducers. When the bacteria are present in low density, autoinducers float away, so the bacteria are unable to perceive them and the bacteria do not produce light. When the bacteria are present in high density, autoinducers are detected by the transmembrane receptor. When the cell population reaches the right density, the lux operon, which is responsible for the regulation of the enzyme luciferase, begins production of the luciferases. The luciferases then catalyze the oxygenation reaction between luciferases and oxygen to produce light. The Vibrio fischeri populate a light organ on the back of the squid's head, where they are fed a sugar and amino acid solution by the bobtail squid. In return, the bacteria's bioluminescence counter-illuminates the squid and protects it from potential predators. Counter-illumination is another defensive function of bioluminescence, where the prey illuminates their body to blend in with the light rays penetrating the surface of the water. This camouflages them with the water surface and protects them from predators who are below them.
The squid is inoculated with the bacteria at birth and develops a light organ on the back of its belly where the bacteria remain. The light produced by the bacteria allows for the squid to mimic the moonlight reflected through the water. The light organ is connected to the circadian rhythm of the squid, and bacteria is able to be produced during the day and then glow at night when the squid hunts. The ability of the Vibrio fischeri bacteria to bioluminesce is governed by a substance called autoinducers. In a high cell population density, the autoinducer is attached to a transmembrane receptor and begins an ATP phosphorylation cascade, which triggers the Lux operon to produce luciferase, and thus light. In a low population density, the autoinducers float away and no light is produced. This unique process allows for Vibrio fischeri to communicate and harbor a symbiotic relationship with bobtail squid.
The first picture towards the top of the screen shows a bobtail squid with its head full of bioluminescent Vibrio fischeri bacteria. The second picture towards the bottom of the screen shows a bobtail squid in relation to its predator. The bobtail squid is above its predator, and the Vibrio fischeri bacteria help the squid to counter-illuminate and blend in with the moonlit water surface. This picture also helps to show the difference between a marine organism that uses the counter-illumination technique with one that doesn't. The counter-illuminated fish on the right clearly camouflages with the water surface better. Looking into the future, bioluminescence is being researched in hopes of implementing some of its aspects into real-world applications. Theo Sanderson is a PhD student at the Wellcome Trust Sanger Institute. He is pursuing the idea of transforming trees into bioluminescent street lamps. Theoretically, this would be done by splicing the genes of fireflies and bioluminescent bacteria with trees. This would hopefully result in a tree whose leaves could provide light along the streets at night. This alternative method of light would eliminate the need for the bulky and inorganic metal street lamps that currently line the streets. Another prospective development of bioluminescence is the idea of smart crops. The idea behind smart crops is to breed bioluminescent crops that could alert farmers to potential disease or nutritional deficiencies through biolumination. Bioluminescence could be useful for use in the Air Force or other military operations. The oxygenation reaction between luciferins and oxygen that produces light is a very effective reaction, with about 98% of energy that is involved in the reaction being released as light. In comparison, traditional filament light bulbs are about 2% effective. These light bulbs also release a lot of energy in the form of heat rather than light. Bioluminescent reactions have a low thermal output. By using bioluminescent sources as light sources, heat-seeking devices would be rendered relatively useless.